Welcome to Olympian Water Testing, giving you the best in water analysis. Today, we are looking at the National Library of Medicine biologic effects of arsenic on plants and animals. As we know, arsenic has long held a position of ambiguity with regards to its activity in biologic systems. In spite of recognized toxicity of many forms of arsenic, various arsenicals have been used in the practice of medicine, and specific nutritional role for inorganic arsenic has been uncovered only recently. That doesn't mean it's all good, though, because animal feeds have been supplemented with growth-promoting organic arsenic additives for many years, and another curious feature of arsenic biochemistry is the ability of the element to partially counteract the ill effects of yet another toxic substance, selenium. So we're taking a look at the toxicity of it. The state and knowledge regarding the effects of arsenic on microorganisms was summarized very well in a study by Mandel et al. On the action of arsenic in Bacillus cereus, trivalent sodium arsenite was found to inhibit growth at a lower concentration, and than prevalent sodium arsenate. The toxicity of the arsenic could be increased by lowering the phosphate concentration of the growth medium, whereas the inhibitory effect of arsenic was dependent of phosphate concentration. This inverse relationship between the toxicity of arsenic and the concentration of phosphate might be related to the fact that arsenic can compete with phosphate for transport. However, DaCosta found that phosphate could suppress the inhibitory effects of arsenic as well as arsenic on the growth of fungi. Mandel et al. Showed that neither arsenic nor arsenate produced any effect on B. cereus and on the incorporation of radioactive precursors into ribonucleic and deoxyribonucleic acids, proteins, or cell wall. Radioarsenate was bound by the microorganism much more strongly than radioarsenate, in agreement with the hypothesis that the toxicity of the particular arsenical was related to the binding of its tissues, because no instance of interconversion between AsB and As3 could be established. It was concluded that the two compounds inhibit the growth of B. cereus by separate mechanisms. The mechanisms of toxicity of arsenicals for this organism seem to be those proposed for mammalian systems, which are later discussed. So what happens with plants? Arsenic occurs in all soils, natures, and waters. Thus, plants have obviously evolved in the presence of arsenic ions. It could, therefore, be that arsenic is an essential element for plant growth but has not been proven, and there are no well-authenticated beneficial effects on arsenic on plants. Arsenic is chemically similar to phosphorus, an essential plant nutrient that can be substitute for phosphorus in plant nutrition. However, it's doubtful in some soils the application of phosphate fertilizer, arsenic toxicity, other interactions between arsenic and plant nutrients are treated later. We can also see the phytotoxicity of organic arsenals. Injury symptoms on crop plants resulting from toxic quantities of arsenic in soils were noted in the 1930s, when it was found that young trees planted in old orchard soils grew slowly and were stunted, so it can have a negative effect on plants. Young apple trees, in addition to being stunted, had leaf symptoms that indicated water deficiency stress, which implied injury to the roots, and peers shown similar symptoms. So we can see all the information there and the interactions between arsenicals and nutrients as well, the mode of action. Now we can also see what happened with lab animals, summarizes the information on toxic and no effect of arsenic compounds. The variety of systems described in the table suggests some of the factors can influence the toxicity of arsenic and arsenic trioxide depend on the purity of the substance. So we can see what's going on here. It demonstrated different species in different resistance to acute poisoning. With arsenic trioxide, mice were less affected by the arsenic compound than were rats. Similar species were shown et al., who noted turkeys and dogs were more susceptible to toxic effects of organic arsenical hydroxide acid than were chickens and rats. Also, we can see here there's different effects on different animals. The work of Harrison revealed that even different strains of mice had different abilities to tolerate arsenic trioxide. These species and strains' differences in toxicity of arsenic could have important implications regarding those of laboratory animals as predictive models for human response. And the average estimated fatal dose of arsenic trioxide for humans is 125 milligrams. Thus, a human is much more sensitive to arsenic toxic effects on a weight basis than a rat. So that's some very good information about how it affects plants and how it affects animals. That's why it's always a good idea to protect them. And one way to do that is getting it out of the air, but another way is just getting it out of the water. That's why it's always a good idea to periodically test your water for any level of arsenic.